Hello, everybody. Welcome you to this webinar series dedicated to application. My name is Luca Clario, and in this section I will speak about Nanolive 3D Cell Explorer Fluo dedicated to nanoparticle studies. The webinar is a recorded version, and is also inside the presentation we see some images that are obscurated with some blank white sheet on it. So keep in mind that the live version is also available on demand. I wish you a good view of the webinar and enjoy. So hello everybody. Let's speak about this application series webinar. Let's go directly with the table of content. The title is the Nanolive Level 3 Imaging for Nanoparticles. As our tradition, we are going to face this kind of application having an introduction about nanoparticles detection principle. We are going to see how the technology is able to detect nanoparticles and what is the inner core of the technique. Despite that, because the series of these webinars are dedicated to application, not going to give you full details about how is the mechanism of the instrument inside. But if you have questions, please don't hesitate to reach us through the website or email for further question or more material. After that, we are going to see different application. First one, we are going to see how the technology can be useful for a first kind of question, for example, how to characterize nanoparticles during the production using a physical parameter that is called refractive index, so optical density number. On the other side, we are going to see some studies about the toxicology, so how to detect nanoparticles and microparticles, for example, also quantum dots in living cells and in tissues. And at the end, we are going to see some application also microbiology. Due to the fact these nanoparticles, they have also effectiveness and in the field of antimicrobial and antibiofilm. At the end, we are going to have a general overview of the benefits of the technique, and we are going to see the future developments. So give me only a few seconds to comment this spectacular first video page. In this case, you see the acquisition done with the latest instrument of Nanolive, so the CLEM. In this case, cells that are going to proliferate directly on the stage in an incubated environment on the microscope. And in this case, you can see how good can be the dynamic of acquisition of the microscopy, high frequency speed, and with the fully high resolution in a large field of view. In this case, in 6 different field of view combined together in a with multiple tiles and in a mosaic acquisition. In this case, you have a good overview of cell interaction, proliferation, and also internal compartment as nuclei and mitochondria. So let's go to speak about in general with this kind of instrument. So those are instruments that are very compact, that you can use on a bench. They don't need even an anti-vibrational table. The one that we are going to focus on is the 3D Cell Explorer Fluo. And the main features are those microscopes are level 3, but they can also use fluorescence to correlate. But the main core technique is don't need any fluorescence marker. They are not toxic because the laser that they are using is really low power. They are able to record the time lapses for a long period of time. up to days or weeks. They are high resolution, comparable to a confocal microscope. They are able to do 3D reconstruction. So in a field of view that is 90 by 90 by 30 micrometers. And they are also quantitative because they behave exactly as a refractometer in 3 dimension, measuring the optical density through a number, the pure number that is called refractive index. How this is possible? Let's see the internal scheme of the microscope. So you have a source of light. This is a laser, a green laser, 520 nanometers. There is a beam splitter, and one is called sample beam, goes through the case to another mirror, and is deflected into this rotating arm here. That is a patented feature of Nanolive instrument, and it's deflected 45-degree angle bend to the sample. It's going to cross the sample completely, and here it is collected by 60 pair air objective, and then it goes down here, and it's compared to a reference beam. This reference beam goes through the case completely unperturbed, and then the 2 lasers combine back together. So because this primary laser is going to be delayed by the fact that it's crossing some biological sample, the 2 beams starts completely aligned, but then you miss the coherence of the phase, and when the 2 lasers combine back together, you create a pattern of interference that generate what we call holograms. The holograms are taken by a camera, and then an algorithm is going to compute back what was the object creating the perturbation. In this way, you can get your reconstruction that is not only 2-dimensional but also 3-dimensional. On this case, you see a reconstruction based on cellular color, following what is the optical density of the cell. And in this case, you have a grayscale representation of refractive index that show you the cells, the staminal cells growing on the stage of the microscope. The final result has 90 by 90 by 30 micrometers dimension field of view, 200 nanometer as a lateral resolution, 400 nanometers as axial resolution, 10 to minus 3 as a refractive index sensitivity. The acquisition speed is around 3D, full 3D in 1.7 seconds. Let's move on. So why the rotating source of light and the 45-degree angle illumination is so important? Why it's so fundamental for the application that we are going to face today? So especially when you have nanoparticles detection, you have distribution of these nanoparticles on the surface, outside the cell, and also inside the cell, probably. So this fundamental feature allows you to have a microscope that is able to generate a 3D automatic 3D rendering, 30 micrometers deep, with everything in focus simultaneously, with no need of Z scanning. So you have to forget about what is the typical scanning mode or focal. You will have with this kind of technology all the Z simultaneously. That is absolutely good when you want to have multiple focal point on Z at the same time. And then you have in this way the capacity to discriminate between nanoparticles that have been internalized and nanoparticles that are on the cell surface. So if you take the example here, see those are map cells infected by Toxoplasma gondii. That is an intracellular parasite. On this case, you can imagine instead of the parasites some microparticles, for example, and then you have all the reconstruction inside the cell in third dimension when you have the parasite or the beads at the ground 0, and then +2, +5, +7 micrometers up on the perinuclear region up to the top part of the cell. So it's very, very good when you want to have an internal localization of these particles. On the other side, having this fundamental feature allows you to expand what is called numerical aperture in a synthetic way. So this feature allows you to expand by 2 times, and expanding the numerical aperture will give you the possibility to collect frequencies that otherwise will be lost. And this allows you to increase what is the high refractive index sensitivity that goes to 10 to minus 3. That is, if you make a comparison, good as a very good refractometer working in 3 dimensions, but in this case with living cells as well. So you have this number voxel by voxel. So you can identify the optical density every single pixel in 3 dimensions. And this is what allows you to have very high contrast, even without the use of any label. So in this case, if you make a representation in grayscale from low to high, dark is low refractive index, high is bright refractive index, you can see here the chromosomes in separation in a cell division, and then all the S reflection of the membrane, for example, or even the mitochondria inside. This capacity to see with high contrast is due to this fundamental feature. And the good part is, as soon you are going to use nanoparticles that have material that is not in the same range of biological material in refractive index, you will get an extremely high signal that is identify the optical density of the nanoparticles inside the cell or outside the cells. Moreover, as you can know, the nanoparticle studies are quite long sometimes. Nanoparticles can take hours to start to interact with the cells, even days to start to penetrate into the cells. So in this kind of study, it's very good having continuous monitoring and being able to follow the cells for a long period of time. That's why these instruments are equipped with a stage top incubator that give you quite a lot of benefits in terms of environment control. So you have the possibility to control temperature, CO2 and oxygen combination, humidity control inside the environment, having also adaptable labware on different format, and also being able to combine everything with microfluidic and perfusion. So in case you want to inject nanoparticles in a different moment, you can also make perfusion, so continue to grow the cells, injecting growing factors. So you have a very good control of your sample inside the chamber. Moreover, he has a very easy access, and also the hypoxia option is available on demand. Here, just the last technical feature before we go for application. So in this case, you have some examples of field of view. In this case, you have on the left the typical refractive index in holotomography. So you have your cells and with some internalized nanoparticles, in this case gold. And this one is the typical grayscale result that you can have. But as you can see on the right, you can have other averages. So in this case, it's rich merge fluorescence acquisition, because this kind of technology is able also to combine with typical epifluorescence. The microscope has an external module, and in this case is available in 3 different configurations: DAPI, pitch, Trich, or pitch, Trich and side 5, or the combination of these two. So you can cover blue, green, orange, and red channels. So it means that not only you have holography, not only you have the optical density to characterize your nanoparticles by a physical point of view, but then you have fluorescence in order to characterize them by a chemical point of view, adding some fluorescence staining. On the right, you have also some parameters. I'm sure you know the benefit of adding the LED illumination. It's very instant on-off, so it doesn't need any warm up or cool it down. It's stable and repeatable. The intensity step is very controlled, so you have 1% step, and the lifetime is very long, over 25,000 hours. So it's really never-ending source of light. But as you can see on the right, there is also a third option. So that one is it's called digital staining. It's part of Steve Nanolive software, and it's a threshold thing that the software allows you to do in order to make a pseudo segmentation of your components of your acquisition based on the optical density. So again, Nanolive gives you the possibility to do this application without fluorescence, with fluorescence, or even with a third combination using pseudo colors in combination with your fluorescence. So you have a lot of different possibility how to study the nanoparticles. We want to move forward. We want to try also to convince you that fluorescence is not the unique way to try to do this application. You need to find, as this reference and happy customer is saying, Nanolive give you the opportunity to read nature with a physical quantity, the density. That's why not to exploit this new opportunity, and why not going also in this direction to make a correlative approach and see also with the density if we can get more information except with simple questions. We also thank the Institut Pasteur in Paris for these excellent images of HeLa cells treated with polystyrene nanoparticles. As you can see on the left, these are very spread cells, fibroblast shape. In this case, you can get the best results with holography. And on the surface, on the side of the cell, you see these granules, these little spots. Those are the nanoparticles. And if you check on the right, on the zoom, you can clearly see how the cytoskeleton and how the reflection and the membrane are going to interact with these little spots. But again, okay, you can have still some doubts, but as soon as you start to do the correlation with fluorescence, the interpretation of holography becomes so clear that we can replace fluorescence in order to see them. We don't need fluorescence, but of course, a multimodal approach can be absolutely beneficial. So let's see the first application that I want to introduce you, that want to respond to some questions like, are my nanoparticles for my study correct? Is this the right choice? Are my nanoparticles appropriate with the correct features for the study that I'm going to do? And that is some fundamental question that we should do before going for a study that involves nanoparticles. So in this case, we have a field of view with glass nanoparticles seated on a plate in some PBS, nothing really complicated, pure sample. And in this case, we have the measure in holography that can generate a 3D TIFF stack on these nanoparticles in 8, 16, or 32 bits. And this will be already sufficient to characterize the nanoparticles in terms of distribution, granularity, and also keep in mind that if you are going to functionalize your nanoparticles with different material, not only their morphology and dimension, but also their optical behavior will change. So ideally, these libraries, they can be used to differentiate between functionalized and not functionalized nanoparticles. Moreover, keep in mind that this morphological characterization is also three-dimensional, so you have also 30 micrometers of Z depth when with 400 nanometers of resolution. So this that I'm presenting you is just one layer of 96 that you can get simultaneously. And as you can see, there is a little evaluation here, linear evaluation of morphology. So those is really matching 200 nanometers linear resolution in lateral view. And on the bottom, you can see the distribution of refractive index between 1.46 to 1.48. So you can also easily get histograms of distribution of optical density. As I told you, from low refractive index dark, high refractive index bright. But on top, if we also use the feature that I explained to you before, so the digital staining of Nanolive, you can make an automatic thresholding and followed by the quantitative software analysis of these nanoparticles. So simply using the optical distribution, I can get a mask like this, having the thresholding of the particles. This will allow me to do, for example, 3D object counter, so determine how many are them, or what is their spatial distribution in third dimension. I can get a histogram distribution, and even more important, because the software knows exactly these red channels, in this moment I can count the number of red voxels that allow me to make an estimation of the volume of these nanoparticles. That is a quantitative measure that will not be absolutely possible using simply epifluorescence. And at the ground, as we are going to see at the end, optical density is correlated with dry mass. So if I know the optical density of these particles and the field of view where they are positioned, I can determine what is the ratio between the two, and using this information, I can calculate the picograms of material that is in this field of view. We are going to see at the end in the application of microbiology. Let's go to the next application. So other particles, as in this case, this batch of microparticles, can be used to study what is the forces that some cells apply to other structures when they are going to do their biological processes. As in this case, they are studied, for example, with macrophagy to study the forces during the phagocytosis, or they are studied with the T cells to the formation of the synapses. And I kindly invite you to go to check some spectacular Nanolive videos of both of these nice applications. So in this case, we are not going to focus on the biological application, but we are going to focus on how these particles to sense force are made. So are those nanoparticles correct? Are those homogeneous inside? Are those homogeneous among them? That is the fundamental question that we are going to answer. So simply measure the optical density, as you can see on the right. If these particles are made by a polymer and you change the percentage of the crosslinker, what happens is if you increase the percentage, these particles becomes more and more compact, and the optical density will increase. So as you can see, if you increase the percentage of the crosslinker, the particle goes brighter and brighter. Moreover, you can use the optical density to perfectly measure what is the optical density connect to the percentage of crosslinker. So as in a batch study, you can simply spot hundreds of those particles and instantly check that all those particles cross-link perfectly and that are all homogeneous each other. And moreover, you can determine what the optical density of each one of them. That is not a non-important information, because knowing the optical density of particles in this case is fundamental for future application in microscopy. For example, if you think to use these particles with a fluorescence study, okay, the distortion that those fluorescence will create depends on the difference between the optical density of the particles and the optical density of the background. Smaller is the difference, smaller will be the optical distortion. So knowing perfectly what is the refractive index characterization of these particles will definitely will be a benefit for you in your decision, and will give you the answer to the question, is this the nanoparticles that I'm looking for? So let's move to the next application, toxicology studies. In this case, we are going to speak about microparticles and quantum dots. Here another reference that I would like to point out, where correctly the professor says the technology is suitable, is absolutely brilliant, and we have to have it because is able to detect the differences in refractive index between the nanoparticles and the cells. This is exactly what we can see in the image here. Those are some human fibroblast cells, and that are treated with titanium oxide. And in this case, you have the nanoparticles that are sitting on top of the cell, and they are forming this multi-star structure that you can see that are pointed out here with the red circles. So around the nucleus, on the periphery, a stick on the membrane. That is very clear observation how these particles are sitting on the cell. So what we can obtain in toxicology studies are really a lot of information. As you can see here, nanoparticles interacting with the surface, also with the vacuoles of the cell, mitochondria, that here if you recognize the nucleus and the nuclear inside. So what we can do is characterize the interaction with living cells and nanoparticles in 4 dimensions, so in 3 dimensions in time lapses. We can have a real-time monitoring of the uptake and internalization process. We can even determine if there is accumulation on a specific cell compartment, and we can localize those particles inside the cell in 3D. Determine if they are on the surface or they are fully internalized. That is really a fundamental question, and is very difficult otherwise answer to these questions. Moreover, we can monitoring living cells morphological changes due to nanoparticles interaction. We are going to see some of this point on the following slide, but before we go there, let me also just point out that we can also spot nanoparticles not only alone, not only in a single cell, but even in tissues. So if we are going to process some tissue, you can go up to 30 microns thick. The best preparation is cryosection, 10 micron thick, better if unstained. And in this case, we have an example of liver tissue with an engulfment of iron nanoparticles that you can see here in the central part of the tissue. And on the right, you can see the pseudocolors based on the optical density distribution. So blue for the tissue, yellow for the iron nanoparticles. So let me give you the first example of this application, checking some interaction between living cells and microparticles. So in this case, we have an excellent embryonic stem cells that is interacting and grabbing microbit that is covered by WNT3 stimulus. So that is in some way reproducing the interaction with the trophoblast cell. And the interaction is really excellent to see. So it's a recruitment, and then the cell is going to orient the angle of division. And it's really, we can monitor how this interaction is going and what is the effect on the cell division due to the interaction with the beads. Not only that, we can also monitor the type, the number, and the maximum extension of the length of each protrusion. And of course, correlative approach using scanning electron microscopy can bring us directly to see this kind of new discovery about what are the estroplexion of the cell, very thin. There's a compartment of the membrane that is called cytonine. And on the same images, it's very evident how this cytoskeletal components is going to interact with the microparticles. So while scanning electron microscopy can give you the resolution, allow you to go in details and see what is the component of the cell in action. On the right, the holotomography can give you the live vision of this and also confirm that the interaction was real. On another study, here we have a different application of microparticles. Before we saw a simulation of some cells with microparticles covered by stimuli. On this case, we have a total different application. is delivery of drug, anti-cancer drug, very famous paclitaxel. And in this case, it is a treatment for lung cancer. Microparticles can be used for carrying this kind of drug directly to the cancer spot. And in this case, are microparticles covered by fatty acid. On the top, you see lauric acid. On the bottom, you see myristic and palmitic acid. And if you check these microparticles with a scanning electron microscopy, you can definitely see that the coating of these particles make them quite different. On the bottom case, you have some delaminated-like microstructures. The microparticles are very porous, and also there is some filament that interconnect them each other. So the researcher wonder if there is any effect of these morphological feature differences to the uptake of these particles from by the cells. And holotomography can answer this question. So if you are going to check the uptake of those particles during the time in 24 hours, you can see that in 12 hours there is a significant difference of the uptake of these microparticles with different coating from the population of cells. And on the 24 hours, this difference will increase even more. So what we can measure with holography, the uptake rate of different particles in A549 longer cancer cells line, and where these microparticles were going to be incorporated and located inside the cell. So in this case, if you make a comparison, fluorescence is not going to give you more information about where these particles, where this accumulation is going to happen. But if you check the optical tomography, it's clear that the control is almost without accumulation of lipid inside, while the other cells are going to make the engulfment of this lipid in the central part of the cell around the nucleus and in the cytoplasm, while the morphology remains completely the same. So they have exactly the same morphology of the control, but with the accumulation of fatty acid in the central part of the cell that is definitely an expression of the uptake of those. As final example of these toxicology studies, in this case we monitor quantum dots. In this case we have fluoroxide quantum dots and cadmium telluride quantum dots, and those quantum dots are used for treating the cells and inducing cell death. That can be different kind of cell death, in general apoptosis and autophagy. So in this case, you can check the untreated cells that is completely flat, while if you check the cells treated with the quantum dots, especially in the nuclear region, in the 3D representation, you see the engulfment, you see the quantum dot accumulation. And here the researchers make also a step further, not only indicating where these particles are going to accumulate it, but also try to analyze how much of them is going to accumulate in the nucleus. So they use the pseudocolors using Steve software. So they gave pseudocolor to the membrane, to the cytoplasm, and then to the nucleus. And they were able to generate the 3D representation. But as I explained you before, you can also make an approach where you can determine the volume of those quantum dots in the nucleus of the cell. So you can, in the graph, you can see clearly that the volume of quantum dots per nucleus in this treated cell is much higher compared to the untreated. Okay, before leaving the toxicology application, just want to share another interesting images from the bioimaging core facility in the University of Geneva in Switzerland. In this case, you have plenty of cells treated with beads. I just want to emphasize the fact that we saw interaction with the cytoplasm, interaction with the nucleus and macro compartment, but it's also good to point out that we can see interaction with the very tiny compartment. As in this case, the question was more if the beads are able to recruiting lysosomes. That is exactly what we see in the central case. So you have the beads, you have this dark vacuole, and you can see here the mitochondria, but then you have these tiny dots white that are the lysosomes that are going to make an aggregation around the beads. So yes, also these kind of applications are completely suitable for the technique. So let's go to the final application. So let's see, we saw tissues, we saw mammalian cells, but we want also to speak about microbiology. And in this case, as you know, nanoparticles, especially silver nanoparticles, are able to release ions that are able to kill bacteria. If you use these nanoparticles also in biofilm, they have antibiofilm properties. So they are not only able to kill bacteria, but they are also able to destroy the matrix in 3D that normally protect these bacteria. So you can see on the left, we have some nanoparticles distribution evaluation. In this case, I really recommend you to follow also our webinar about transmission electron microscopy to determine the morphology of those nanoparticles and to characterize their from the morphological point of view, analyzing the dimension, making distribution. We have another webinar dedicated to that. This is normally the approach that you do in transmission electron microscopy. But then the scanning electron microscope can give you the confirmation that those nanoparticles are really on the field of view and they are starting to interact with the bacteria. So you can start to see the bacteria and the nanoparticles stitching on the wall and destroying the wall of this bacteria, inducing the explosion. So excellent. We have also this. So we select correctly the nanoparticles. We know that the nanoparticles are killing bacteria, but how many of them are going to be killed? So in this case, we have the first application of holotomography. You can simply make an acquisition in a field of view where you have nanoparticles and bacteria, and thanks to the pseudocolors, you can check the surfaces, and instantly all the bacteria will be marked with your pseudocolors. So instantly you can have the number of them, and even better, the volume of them in 3 dimensions. So you can make an estimation of how much percentage of your population the nanoparticles are killing. So that is the first application that your holotomography can guarantee you. And the second case is the effectiveness on the biofilm. So the biofilm is in 3D, and knowing exactly how percentage of the biofilm is going to be destroyed with the nanoparticles is not easy at all. Because, for example, if you use crystal violet to make this estimation, crystal violet is absorbed also by nanoparticles. So crystal violet is not a good approach because it's going to give you a lot of false results based on the fact that nanoparticles are absorbing the color. So you can mislead, kill bacteria or destroy biofilm by the fact that the crystal violet is absorbed by the particles itself. So in this case, the researcher used the holotomography to define what was the total volume of the biofilm, and then after the treatment with the nanoparticles, you see that the biofilm disappeared completely. So again, quantify biofilm dispersion and then the reduction of bacterial cell number. Final point, as I promised you before, I'm going to speak to talk to you about the dry mass estimation. So in this case, because we are speaking about microbiology, what is dry mass in this case is the composition of the organism in if you exclude water. So what is the weight of this bacteria if you take out the water? In this case, is DNA, RNA, lipid, but main part of the organism is protein. And indeed, when you measure dry mass, it's everything together. So you have a precise number of the picogram of material of this kind of macromolecules. And why is important knowing the biomass, or why is important having the dry mass? In this case, when you are studying T cells and you know their biomass change when they become specific. When you study stem cells, when a colony change their the properties and they're going to differentiate on something, they change the biomass. And as well, if you apply to hemoglobin to the red blood cells, you can definitely determine the hemoglobin concentration through that. And also really recent application demonstrated you can even study and determine cell death processes using the biomass change during the time. And if you apply in bacterial growth, you can determine how much biofilm and 3D colonies are growing or being destroyed. So what is the connection? You can see it here. So you can determine dry mass having the refractive index of the sample, and this is the measure that the microscope does for you. And this one is the refractive index of where they are immersed. So in this case, with water, but then you can change with the refractive index of the solution that you are using to cultivate your cells. So it's very easy to obtain this number, and it's very powerful. And another application that I can suggest to you is the measure of the quantity of nanoparticles uptaken by a cell, also through this approach. Here we are at the summary of benefits. So as we saw, what we can do with the holotomography, we can do a level 3 characterization of nanoparticles. We can do a long-term high-resolution monitoring of uptaking and internalization, of course, starting also the kinetic and dynamic. We can determine the 3D localization inside cells or on the surface. And we can see what is the morphological changes of these nanoparticles induced to the cell. And also I remind you that we can calculate volume and dry mass, and we can do that in a specific compartment or even in a tissue. So I hope that was clear for you. And if you have questions, don't hesitate to reach me directly or through the Media System website. We also remind you that we have a demo unit, so we can also, if you are interested to see some of your sample, we can provide demos. And definitely, I suggest you, encourage you to follow what is our webinar series with the next chapters. Last news, probably, we have also a new partner, DR Vision, that can provide also software dedicated for machine learning and deep learning to do object detection and object tracking in B dimension and 3 dimension. That will be absolutely useful when you try to track the nanoparticles inside living cells. We want to thank Nanolive for the material and for the support for this webinar. And I thank you, all of you, for your attention. I hope the webinar was interesting, and I would like to see you on the next appointment. Take care and see you next time.